Coming to the next type of asexual reproduction is budding. Budding is nothing but a tiny outgrowth or a protrusion which can be seen from the parent body. Depending on the presence of where this outgrowth can be seen, there are two types of budding: exogenous budding and endogenous budding. In exogenous budding, the bud can be seen on the outer side of the parent body, whereas in endogenous budding, the budding or the bud can be seen inside the body of the organism. So, exogenous budding examples are yeast, which is a unicellular fungi, and hydra. Here is an hydra which is having a bud on the outer side of its plant body. So this is nothing but an exogenous bud. This bud, when it gets detached from the parent body, it has the capacity to develop into a new individual. So exogenous budding is nothing but the budding which can be seen on the outer part of the parent body, and when which detaches from the parent body can further develop into a new individual. Whereas in endogenous, the bud can be seen inside the parent body. This is a spongilla. This is the only fresh water where you can see endogenous buds. The buds here, or the gemmules as we say, are seen inside the parent body. These gemmules, when are completely formed, comes out, or it is getting released into the environment, which on a suitable substratum falls and forms into a new individual. When you take the individual gemmule and its structure, you can see a centralized mass of cells which is surrounded by a cyst or a protective layer. In this protective layer, you can see needle-like structures which are called as spicules, which are responsible for protection. You can also see a micropyle, which is the opening. So basically what happens is, when the gemmule comes out of the sponge, when it falls on a suitable substratum, from the micropyle it grows and develops into a new individual. After budding, the third type of asexual type of reproduction is spore formulation. The spore formulation can be seen in non-flowering plants or some protozoas. Spores are nothing but small round-like structures which, on falling from or away from the parent body, can develop into a new individual. Basically, there are two types of spores: zoospores and conidia. The main difference between these two are zoospores can be seen in algae and conidia is seen in fungi. Zoospores are basically motile. If you take Chlamydomonas as an example, these Chlamydomonas zoospores are biflagellated, meaning it has two flagella, which is basically required for movement. So, which is, the zoospores are nothing but they are motile. They can swim around in the water, basically in the water medium, and which is seen in algae, which consists of flagella, which is helpful for movement. Whereas conidia can be seen in penicillium or the fungi. These are non-motile spores. Basically what is conidia is, these are nothing but chain of spores which are attached to one another. As you can see on the fruiting body, you can see the chain of spores which arises, and when it detaches from one another, it falls on a suitable substratum and develops into a new individual. Coming to the next type of asexual type of reproduction is fragmentation. Fragmentation is nothing but the cutting of the fragments or the pieces of the body of the parent. The parent body here splits or cuts into different fragments. It can be regular or irregular. On fragments, on formation of fragments, each fragment has the capacity to develop into a new individual. This fragmentation can happen once or twice. This is seen in Spirogyra, which is a blue-green algae. We also have an example of Planaria, that is in animals, Platyhelminthes. As you can see, there are different fragments and at different points of the body. This each part has the power for regeneration, meaning it has the capability to form into a new individual. So basically, fragmentation is nothing but the fragments or the cutting of the filaments of an organism which has the capacity to grow into a new individual. Coming to the next type of asexual reproduction is the plasmotomy. This is the last type of asexual reproduction, and the next type that we will be seeing, it will be in plants, that is basically in vegetative propagation through stem, leaf and roots. So plasmotomy is nothing but a stage, or which can be seen in some protozoas, where in a multinucleated organism it can be seen where the individual cell, when we take the individual cell, usually in a normal type of cell, the division takes place usually with karyokinesis first and then cytokinesis. That is nothing but the division of nucleus first takes place, followed by the division of cytoplasm. But here the organism undergoes cytokinesis first, that is, the cytoplasm gets divided first, later the nucleus divides. So as you can see in the parent cell, the cytokinesis takes place, that is, only the outer is getting divided, and this can be this individual cell's nucleus that you can see can be divided equally or not. So we can see the cytokinesis first, where there is only division of cytoplasm. Later it will get divided through nucleus. There is nuclear division which undergoes mitotic division, which results in doubling of each individual cell. This can be seen in a plasmodium, which is also a malarial causing parasite. So this is about the asexual type of reproduction, and we studied about what is reproduction, what is life cycle and types of reproduction, and we studied in detail asexual type of reproduction. So basically in asexual type of reproduction, we study from fission, which is of two types, binary fission and multiple fission. We studied about budding, which is also of two types, exogenous and endogenous. So based on the outgrowth which can be where it is present, we can place them as endogenous or exogenous budding. We also saw about spore formulation. We studied about zoospores and conidia. Zoospores are seen in algae, whereas conidia is seen in fungi, especially in penicillium. And Fourth type was the fragmentation, where a plant body cuts into individuals which can regenerate into a new plant or a new organism. Later, we also saw about the last one was the plasmatomy, where the multinucleated cell usually divides with cytokinesis first and then undergoes karyokinesis. Example is Plasmodium, which is a malarial parasite. So, this was a general account of asexual reproduction in single celled organisms. In the next session, we shall take about asexual reproduction in plants in particular.